Right. So Sylwia is up next, and she's from the University of Southampton. She will be discussing some exciting work on hyperpolarized 113C fumarate on a microfluidic chip.
Hi, thank you very much for the kind introduction, and just let me know if you cannot see my screen.
We can see the screen.
Yeah, just a second because I have a small lag.
Yes, there we go.
Okay.
Perfect.
We're in a full screen right now.
Yeah.
Okay, so thank you very much for inviting me here to present our research, and our motivation behind this work is to produce a hyperpolarized metabolite by FIP at a microscale. So our group in general specializes in performing chemical reactions and biological cell culture at microscale. So here I'm just showing you a couple of designs. The first one is a gas liquid. The first one is just for gas liquid reactions, which I will discuss in details in the remainder of the talk. Others allow us to perform peristaltic pumping or droplet generator. And all of the functionalities are allowed by just a clever design of these microchannels. Now, with all of the functionalities that are possible, it's important for me to highlight that our devices are biocompatible, and we can perform tissue and cell culture inside of the magnet and observe real-time metabolism. So implementing hyperpolarization of metabolites will allow us to access metabolic pathways which are otherwise unobservable due to limited sensitivity. So the reaction that I'm going to discuss today is a reaction of parahydrogen with acetylene dicarboxylic acid disodium salt in the presence of ruthenium catalyst, and that results unusually in a production of a transhydrogenated product, fumarate. And so fumarate protons are chemically and magnetically equivalent, unless there is a carbon label present, which allows us to break that symmetry and access the singlet order. So we've done that by using this S2M pulse sequence. So generally, people bring in just conventional NMR experiments, the solution is brought in contact with parahydrogen by means of bubbling or shaking. And bubbles pose difficulties in microfluidic devices because they alter the flow properties and sometimes even block the channels. So what we've done is we used a semi-permeable PDMS membrane. So that allows us to, so the membrane allows for the gas to dissolve into the liquid interface. And now all of our experiments are performed in a high field NMR spectrometer, and the gas the gas is delivered externally from the magnet, just from a Bruker parahydrogen generator, and the precursor solution is delivered using syringe pumps. And inside of the NMR spectrometer, our chip sits in our home-built transmission line probe, which ensures planar orientation of the chip. And here we have our chip holder. So the chip holder holds the assembled microfluidic device together with the PDMS membrane. What it also has is just these threaded nanoports, which align with the channels on the chip and they allow for the delivery of substrate and the gas. So the reaction is performed in 50 degrees, so elevated temperature, and we use that by we've achieved that by clamping a heater here on the chip, so it only heats up the sample chamber, whereas the top of the chip, where the hydrogen dissolves, stays cold in order to enhance the amount of hydrogen that we actually can dissolve. Lastly, our chips are positioned in the stripline plane in this region, which I highlighted here. So that's where this stripline plane concentrates the RF field, and that's where we observe all of our substrate products, sorry. And so this is the top view of the chip. So the precursor solution is delivered in the blue channel right here. And the hydrogen is delivered into the red channel, and the green signifies the PDMS membrane. So hydrogen penetrates the PDMS membrane, which just acts like a bridge and dissolves into the solution channel. So here are our results. The Fumarate peaks are shown in blue. And if we compare an integral of a single scan hyperpolarized spectrum with 400 scans of a reaction performed with thermal hydrogen, we get the enhancement factor of 190. And just to emphasize that all of our experiments are performed at steady state flow at 10 microliter per minute. And now enhancement factor of 190 is quite low, since with similar reactions, we're able to obtain at least 10% of proton polarization. So if we just examine the hyperpolarized spectrum further, we can see that there is this peculiar feature of a partially negative line, which tells us that something else is going on in the reaction. So it is known that hydrogen molecules and heavy metal catalysts form intermediate hydride species, where the two hydrogens take non-equivalent positions. And if the residence time in such complexes is long enough, the nuclear singlet state will leak into the triplet state. So this is called singlet triplet mixing, and it was already discussed at this conference in the context of SABRE yesterday, and I think the day before as well. So in the ideal case, we have no population in the triplet manifold, and the S2M pulse sequence will give us this beautiful sharp peak. But where a singlet triplet mixing occurs, there is a population in the triplet state. And so although the S2M pulse sequence just acts simultaneously on all of these states, if we look at them just for simplicity separately, the singlet state and the triplet state, they destructively interfere after the pulse, the singlet S2M pulse sequence, and that results in a decreased peak. So there were a few methods already discussed at the conference, and today we will also use similar methods, which is just the CW irradiation. So by applying CW irradiation, we scrambled populations equally around the triplet manifold, but most importantly, we can prevent the singlet triplet leakage if we are on resonance with the species and the power of the CW is strong enough. So here we have performed this experiment, and we applied a mutation frequency of 500 Hertz and varied the resonance offset from 20 to -20. And we can see that at around -7, we have a increase in signal amplitude by threefold. So such a picture isn't very uncommon for Saber experiments, but for hydrogenated FIP, these are much harder to acquire, as each of these points would require a new sample to be prepared. So these experiments we've performed in only 30 minutes, and we used only 400 microliters of the sample. And as I said, the signal amplitude increased by threefold, giving us about 600 enhancement, which is 2% proton polarization. So applying CW radiation is not an optimal solution for the chip, because the radio frequency field focuses just at the sample chamber, whereas the entire reaction, whereas the hydrogenation reaction takes place just upstream from the sample chamber as well. And for this method to be effective, you really want to irradiate throughout the entire reaction. So now another method can be used, which is just applying a hard pulse between before this the pulse sequence, which will essentially move the population from the T0 state to both or outer states in the triplet manifold. So now to investigate that, we varied the pre-pulse from 0, so just a regular singlet to S2M pulse sequence, all the way to 2 pi. So as we increase the flip angle, we can see a steady increase of the signal enhancement. So on this axis, we just have signal enhancement, and we have a maximum at 90 degrees, which corresponds to 6-fold signal enhancement compared with S2M pulse sequence. And as we go away, this enhancement decreases just as expected, and then again going all the way to 360. So now comparing just the S2M pulse sequence to 90 S2M, we see a nearly 6-fold increase in the enhancement, and that means that there's a lot of singlet triplet mixing occurring on our chip in the reaction. So to conclude, we have produced a continuous stream of hyperpolarized fumarate by FIP in a microfluidic device and achieve 4% proton polarization. The stable hyperpolarized stream allowed to study singlet triplet phenomenon in hydrogenated FIP, and microfluidic chips provide a fast and accurate platform to study physical phenomena. And just to go back to my initial slide, so we have produced a hyperpolarized metabolite in water, which is great. But in order to make it compatible with our cell cultures, obviously the catalyst and the remaining starting materials need to be captured, as well as the temperature of the reaction needs to be reduced, because that will unfortunately kill the cell cultures. But we are on the right track. So with that, I would like to thank my wonderful team at Southampton and collaborators as well. And thank you very much for your attention.
Awesome. Great talk, Sylvia. The microchips are so cool these days. Just curious about that. How expensive are these microchips?
So actually, depending really what you are counting as a cost, because the probe, obviously you need a specialized microprobe. So if we build the probe, is less than €1,000 for us. And one microfluidic chip, less than, I think I might have lost connection.
We have
Sorry,
you.
I think I was having some connection error there.
Yes. Oh, I'm back, I think. Yeah, I've lost connection.
Okay, we got one more question. So were you able to measure singlet lifetimes?
Well, we're in flow, so that would be, yeah, no, we can measure singlet lifetime if we'd like to do so, but we haven't done it now.
Okay, and then we have one more. Do you expect that if you could decouple the protons throughout the process, that it would approach the enhancements seen in the batch process?
Yes, I think so. Yes. I think just the CW irradiation in theory should suppress singlet triplet mixing. So yes, it should do so.